Okay, so the next speaker is Ayden Kıdırmanov, who will be talking about studying the virome of Caspian seals. Ayden, are you ready?
And hello, everybody. So we study Caspian seal virome, Pusa caspica virome study. We've been implementing this project for 2 or 3 years already. We monitor the status of marine mammals population. This is one of the priorities in our studies. And this is an integral component in conservation of wildlife and control of potential threats to marine mammals and humans. The main infectious diseases affecting marine mammals, including Caspian seal, well known adenoviruses, the viral infection, calcivirus, herpes, influenza virus, and morbillivirus, pox virus, and Lissa virus, ephemera virus, vesicular virus, and coronavirus. And a specific role is played by bacterial infections, actinomycete, brucellosis, Clostridium, pneumonia, and infectious diseases, leptospirosis, streptotrichosis, mycobacteria, tuberculosis, and mycoplasma in animals. And the previous studies of infectious diseases and mostly focused on the bacterial disease investigations. It was related to the permanent stranding of Caspian seals. Different years. The first stranding documentary confirmed it was done in 1932, Tюленей Island. We found 6 corpses, carcasses within 100 meters along the seashore, and mass die-off was recorded in 55 and 56 of 20th century, and around 30,000 animals died, and it was a diplocoque infection, but I should say that perhaps it was a secondary bacterial infection, and back then we didn't have a very good virology scientific base, and classical methods. We were not able to come to the true reason. Bacteriological infections are easier to detect and determine, and we thought the pathogens act as a primary vector of diseases. In 1964, also around 40,000 animals died without any reason identified. In 1968, around 3,000 animals died due to geological survey and seismic survey, and the biggest die-off of animals occurred in 1997, the year of 2000. And it spread across the entire Caspian Sea and across the entire water surface of the Caspian Sea. In Kazakhstan, they counted more than 10,000 dead seals. I do not really know. From my perspective, we counted also old carcasses, and even old carcasses were considered and included in the total number of dead animals. And similar research of the mass die-off of the marine mammals, and it was delegated to veterinary labs. and they just were able to identify bacterial infection typical for agricultural animals, and viral infections were not diagnosed. And even now it's a problem, and back then it's even so. And they have a limited range of capabilities for diagnosing viral infections. Out of viral infections, mostly they tested influenza virus in the end of 70s, beginning of 80s. We started viral infections, and in connection with the fact that in the beginning of the 80s there was a mass die of seals in the United States, and from respiratory infections. And as a result, the influenza A virus was isolated from the dead animals, and it gave impetus to study viral infections among marine mammals. And virologists that are dealing with the influenza viruses, they started to consider other viral infections from the environmental impact perspective. And in the former USSR, starting from 1976 and up to 1999, Svetlana Yamnikova studied samples taken from 152 individuals of Caspian seals, but back then she didn't find any confirmation of the viral vector origin, and also serology study on antibodies detection to influenza viruses were done, and electronic data, and also there was a data on the presence of antibodies to influenza virus A and B, both types, population of Caspian seal, and they proved they had a circulation of epidemiological strains of the. Yeah, you have 5 minutes to go. And here you can see the research performed by other virologists. And apart from the influenza virus, also the plague virus, and there was a mass die-off of marine mammals, pestilence, and also other vectors. We performed over the course of 10 recent years. We gathered materials from different species virology studies, and as a part of virology studies, we managed, along with the serological studies, we identified, here you can see virology study data and the presence of the virus. And this is the pestilence virus, and the influenza A. And in many animals, the distemper virus and the herpes virus were determined, and serological studies were done, the circulation of viruses among the Caspian seals, influenza A, influenza B, adenovirus, coronavirus was also found in the seal populations, and also antibodies to morbid viruses. Here you can see the chart, the table showing serological studies. And these are the studies performed to classify the influenza virus, whether it's A or B subspecies and substrain. and mostly they had A with the subtype DH17, and also there is a B influenza virus, and we also studied viruses, and we started to apply most advanced techniques like genome sequencing, and we detected different groups of viruses. And in the diagram you can see the viruses, and high homology was detected to flaviviride, retroviride, coxviride, and pneumoviride, and also had the samples that are similar to avian viruses, but we need further confirmations that the viroma viruses, perhaps they have an alimentary origin related to the food chain, and the leeches of blood-sucking insects, fish, and seals. And the analysis will enable to identify the degree of infection among the Caspian seals and better classify the viruses. The virome of Caspian seals provides a full spectrum of information on the vectors, enabling us faster identifying infections and regulating their spread. Here you can see the takeaways on recommendations and the spectrum of infectious diseases affecting seals in the Caspian Sea. Thank you. I'm done.
Thank you very much, Aydan. I think in order to keep to the conference schedule, we should move on to the next speaker now. Perhaps there'll be time to ask Aydan a question at the end of the session.